Amherst. He then went on to do a PhD with Pat Schloss at Michigan, studying the microbiome in cancer. And then as a postdoc, he moved down to Vanderbilt, where he studied the microbiome in the context of pathogenesis again. So he started his lab at CHOP, where, as you will see today, he's been working feverishly on understanding the role of the microbiome in C. diff infections. He just published a really big paper just a few weeks ago, less than a month ago, and I'm sure we're going to hear most of that story today. It's really, really beautiful and elegant. One of the things that I love about Joe's work is that it's incredibly comprehensive. He really attacks a problem from all angles. There is no technology approach, anything that is off the table. He just kind of attacks the problem with everything that he's got to get to what the answer should be. So I'm really thrilled to have him here, and I hope you enjoy hearing all about C. diff infections. Thanks, Joe.
Thank you so much, Emily. That was a really generous introduction. If you want the slides, I can afterwards. I'll email them. Happy to. Thank you all for coming. It's really fantastic to be here. I actually last time I was here was when I was I think it was an undergrad with Pat Schloss at UMass, and we were. Mother had just come out, if anyone has ever used Mother, and we were going around doing workshops, and we came to Penn State. I ate a ton of ice cream at the creamery, probably still haven't got rid of those pounds, and I'm trying to figure out how I can get ice cream back home with me, what my wife has demanded. So if anyone knows how to.
You can also buy online and they ship it to your house.
I know, and that's a little worrisome. For me,
they sell dried ice there.
Yeah, they sell dried ice. That sounds amazing. Okay, but I'm really delighted to be here. I'm excited to tell you about the work that we've been doing in my lab. As Emily noted, I started my lab at CHOP in 2018, and we've been working on this project focused on the pathogen Clostridioides difficile that I think a lot of you are quite familiar with. Here we go. So C. diff, this is just the cartoon that we always put up for C. diff. This is the CDC's 2019 update on the antibiotic resistance threat report. And the CDC classifies C. diff as a threat level urgent. That's because C. diff causes hundreds of thousands of cases every year, over 10,000 deaths a year. And it's a huge problem in terms of the burden it causes on the healthcare system. These numbers are actually thought to be pretty underestimated. The CDC did a trick where they focused primarily on just healthcare associated infections here, but we know that there's been an emergence of community associated C. diff infections recently, and so we think that these numbers are obviously much higher. But as a microbial ecologist, I've always been really fascinated with C. diff infection. I don't have to tell this group, but microbiome is really, really important for our health, right? But I think C. diff is one of the best examples of the power of our microbiome. And so under normal conditions, we're all resistant to C. diff. And so, I work at the Children's Hospital. We're at the University of Pennsylvania. There's lots of C. diff spores around, but we don't get infected. We're largely resistant because of colonization resistance that our microbiota confers. Probably the best way to exemplify this, it always fascinated me, is if you go down to the animal facility at Children's Hospital Philadelphia, you can gavage in billion spores of C. diff and nothing happens to the mice at all. They're totally fine. We can't even get colonies out the back end. However, if you give those mice a broad spectrum antibiotic or a cocktail of antibiotics in the same strain, a virulent strain, all those mice will die. And so it really shows how well our microbiome is equipped to preclude organisms like C. difficile from colonizing. However, when this microbiota is perturbed traditionally by antibiotics, it's the #1 cause of antibiotic-associated diarrhea, C. difficile spores from the environment can colonize. So we know that the main germinants for the spores are primary bile acids. So enterococci is one of the best germinants for C. difficile, and C. diff will come out of its spore after it sees these primary bile acids and produce its two main virulence factors, TCDA and TCDB, or toxin A and toxin B. I always find it really fascinating because C. diff, unlike a lot of the other spore forming, like the bacilli, have a really good barometer to know when they're in an environment that's conducive for their outgrowth, right? Because they don't just come out of their spore when they have nutrients around. They can kind of sense the metabolic chains that are disrupted during antibiotics that then come out of their spores and cause disease. And so one of the observations that's always had me interested and kind of drive some of the work, most of the work in our lab, is that when somebody gets C. diff infection, so say they're at the hospital, the University of Pennsylvania, we see a really broad spectrum of disease manifestation. And so even in adults, we can see asymptomatic carriage in some of the patients in the hospital. Some patients will get mild diarrhea, but then some patients will go on to really, really severe manifestations of C. diff infection, like pseudomembranous colitis, toxic megacolon, or even death. What's really interesting is that this doesn't really seem to be driven strictly by the strain of C. diff. And so, back, 10, 20 years ago, we thought that this was largely driven by potentially hypervirulent strains of C. diff. But even if we go in and we do genome sequencing and looking at pathogen surveillance, what we really find is that there doesn't seem to be an association with the strain itself driving this. And so, somebody in room A has a lot of the same risk factors, the same strain of C. diff as somebody in room B. However, the person in room B gets really, really severe disease and dies. The person in room A doesn't really seem to get that sick. They can clear with antibiotics. We don't really understand what's driving that spectrum of disease. Additionally, there's this paradox in C. diff infection, because what causes C. diff infection is antibiotics, right? And that perturbs the microbiota. And then what we treat somebody with C. diff with is antibiotics. And so by definition, when you're treated for C. difficile, you're becoming susceptible to C. difficile. And so what ends up happening is that somebody that gets a C. diff infection has a really high rate of getting reinfected or recurrent infection. So around 30, 40%. And so if you get a recurrent infection, you then have an even higher risk of getting a subsequent recurrent infection again. And so it goes up to like 60, 70%. And so what ends up happening is that patients get in these debilitating cycles of reinfection with C. difficile. And so what will happen is they feel really, really sick. They'll go on vancomycin. As soon as they take off the vancomycin, the C. diff will come back. And so that's kind of where the idea of fecal microbiota transplant has come in. I'm not going to go into that today. There's some really amazing work being done here, but it really shows that there's this kind of paradox with C. difficile in this really challenging phase where our main tools to combat this infection cause the infection itself. And so kind of thinking, like stepping back, this kind of, we know that the strain doesn't really seem to be associated as much with driving the infection. We don't really know what's driving the spectrum of disease. And so that, brings us to the hypothesis that there's largely other environmental, microbiota, and host factors that are kind of driving this disease manifestation. That's kind of where we've come in my lab and become very interested. And so if you think about enteric infections or infections of the gastrointestinal tract in general, we know that it's really complex, right? So you have not just the invading pathogen, but you also have the interactions with the pathogen and the host, the immune response. You have this really complex microbial community that's there. In the sense of C. diff, this is a perturbed microbial community that might be rebounding from antibiotic treatment, so it's really dynamic. We know that the microbiota and the pathogen can interact with each other. We also know that the microbiota can really influence the immune system as well. So what the host is doing can really be influenced by the microbiota. And I think a lot of people in this room are also looking at and know how complex this actually gets when you start to think about things like diet and dietary factors, as well as drugs and supplements and things like that. And so it's really, really complex. And so, when we're studying these things, and everyone in this room are studying these communities in the gastrointestinal tract specifically, you almost have to just ignore the complexity, embrace the complexity, and kind of drive into it. But it can be really hard when you start to think about molecular mechanisms, what exactly is going on. And so in the lab, we're very interested in, all of these aspects. We're doing a lot of work and looking at the host and vaccine development. We're looking at how pharmaceutical drugs, specifically, we've been working on NSAIDs, impact this cycle here. And in the past, we've done some work on diet. But today I'm going to tell you a story looking at how microbes are influencing severity of C. diff infection, specifically the Enterococci. And this work was largely driven by Alex Smith, who is a Penn State alumni. Many of you might know Alex. Really phenomenal graduate student in the lab in the microbiology program at Penn. And so one of the things that has always interested me is the fact that if you look at, so I guess I'll step back just for a second, which is that when you think about, C. diff infection, it's invading this polymicrobial space, right? And that context is really, really important to what C. difficile is seeing and what might be influencing its behavior. And so we were really interested in kind of understanding how that context, how the microbial context impacts the infection itself. And if you look at patients with C. diff, one of the things that's interesting, so this is children at the Children's Hospital of Philadelphia. This is just metagenomic sequencing, looking at different taxa with bar charts, and then after the various visits over time, after they were diagnosed with C. diff and treated with antibiotics. But what you find is just like during health, how we are all very diverse in our microbiotas, right? We all have different microbiomes, but we're all healthy. When you look at the C. diff microbiome, or what's happening when people are perturbed and they get C. diff infection, we actually find just as much diversity, right? So in general, we find that people with C. diff, they have correlates that are associated with C. diff infection and susceptibility to that infection, but they're largely very different, right? And so this kind of context by which C. diff invading can be very different from person, even though all those people are susceptible and have been perturbed, that kind of perturbation can be very different. And so we were very interested in kind of understanding some of these associations, which organisms might be driving susceptibility to C. diff infection. And there's been a number of studies that have been done, kind of looking at this in a survey, in a microbiome survey way. And so this is work from Eric Paymer's group. This is work from Pat Schloss's group. And the Paymer group was really focused on trying to understand some organisms that might be useful for antagonizing C. diff. But what you can see down here is that they also find a lot of organisms, or some organisms here, specifically Enterococci we'll focus on, that are positively associated with susceptibility to C. diff infection. The Schloss lab did some work where they took mice from various different sources. So, his own colony, Vince Young's colony, Jackson, Charles River, Taconic, and they looked at how C. diff was impacting these different environments, these different starting microbiotas. And what they found is that the length by which C. diff is excluded from the microbiome after infection really is linked to where you get the mice. This is really scary, right, everyone? We get mice from different sources, we might be trying to reproduce our stuff. It can really matter where you get the mice and what the microbiome is. But one of the things that they noted is that the mice that carried C. diff for longer periods of time tended to have more Enterococci. And something that I've never really understood, because I was at Michigan when they were doing a lot of this work, is that Vince Young somehow has created a community that doesn't have Enterococcus. We can't get rid of Enterococcus in our mice, so I don't know, I'm still trying to figure out his secret. But we also did some work where, when I was in Eric Scar's lab for my postdoc, we were looking at how dietary metals impact C. diff infection. And one of the things that we were working on is dietary zinc. And what we found is that when you gave mice excess zinc, you altered the microbiota and enriched for the Enterococci. And what this did was this associated with an increase in susceptibility to the infection. So if you give these mice very, very low levels of antibiotics, the excess dietary zinc and then enrichment of Enterococcus allowed C. diff to colonize where normally it couldn't. And it also dramatically increased disease. We also found that the Enterococci, they bloom during this excess zinc phenomenon in the dietary intervention, and then they also, it leads to complex disease, and then the Enterococci, they get out of the gut and cause bacteria. And so we were seeing all of these associations in the literature and our own work where Enterococci was associated with increased susceptibility and severity of infection. And so we became very interested in trying to understand how the Enterococci might be playing a role during C. diff infection. And so we wanted this was consistent in our own patient cohorts. And so we had a cohort from Vanderbilt and a cohort from the Children's Hospital of Philadelphia. What you can see here is that if you look at patients with C. diff, they tend to have a lot more Enterococci in their gastrointestinal tract during C. diff infection. And if you look at the amount of Enterococcus correlated with the amount of C. diff, if you have more Enterococcus, you tend to have more C. diff, and this is positively correlated. And so additionally, if you look at adults at the University of Pennsylvania, if you have more Enterococcus during a C. diff infection, you also have higher metrics of disease severity measured by with white blood count. And so this all is pointing in the direction of associations between this one taxa and severity and susceptibility to C. diff infection. And so what are the Enterococci? The Enterococci, I think, are really, really fascinating organisms. They are one of these classical friend or foe members of the microbiota. So all land animals have Enterococci. So you can find Enterococci in birds and lizards and humans, and they're members of the normal healthy microbiota. However, they're also really important pathogens. And so Enterococci are intrinsically resistant to a lot of antibiotics. They're also able to become vancomycin resistant. And this is when they can become a real challenge in terms of the healthcare setting. And so this is again the CDC report, and you can see that Enterococci are a threat level serious for in terms of their challenge in the healthcare setting. And so Enterococcus faecalis and Enterococcus faecium are the two most important for human health, but there are a lot of different Enterococci. And so additionally, the risk factors for VRE completely overlap with C. diff infection as well. And so thinking about antibiotic treatment, the length of your healthcare stay, your age are all positively associated with having VRE. And so we were very interested in understanding if this was a correlation or a causation. So we know that the Enterococci, they're like weeds in the gastrointestinal tract. When there's a perturbation, they do really, really well. They grow to really high abundances. So is this just an association with C. diff, right? So, they're both, and it's antibiotic treated, it's dysbiosis, they thrive. Is that just almost like a biomarker of C. diff infection, or is it actively impacting C. diff infection? And so we wanted to know, do the Enterococci actually impact susceptibility and severity of C. diff infection directly, or is this just a causation? So one of the first things that we did was we moved to our animal model of C. diff infection, and we used different antibiotics to kind of tune the amount of Enterococci that are in these mice. And so the normal model that we use is the one that was developed at the University of Michigan by Casey Terrio using broad spectrum cephalosporin, cephalosporin. And this makes mice susceptible to C. diff infection. It really alters the microbiota. But what's important to note is that Enterococci are intrinsically resistant to cephalosporins. And so they do just fine during this perturbation. You really disturb the community, but the Enterococci thrive. And alternatively, if you add in vancomycin, and so normal commensal Enterococci do not have vancomycin resistance, so you can deplete the Enterococci in these mice. And so this is kind of showing this, the data here in this schematic, where if you use vancomycin with cephalosporin, you can completely deplete the Enterococci during C. diff infection. And so what we did was we introduced C. diff in these different conditions, and what we find is that when you use cephalosporin, so here's in the black is C. diff, and then this is commensal Enterococci here in the red. You can see that C. diff colonizes just fine when there's a lot of Enterococci around. But what we noted is that when you deplete the vancomycin from these we use vancomycin to deplete the Enterococci from these mice, we saw a defect in the ability of C. diff to colonize early on. And so what's important to note is that the Enterococci, as I said, are really hard to get rid of. And so they come back pretty quickly. And so there's this window where the Enterococci are depleted from the vancomycin, but they're coming back and they're really, really low abundance. And if you add C. diff at that time, C. diff has a defect in colonizing. However, eventually the Enterococci come back and then the C. diff will come back up as well. But what we can do is we can add in exogenous Enterococci from the lab at this time point. And what we found is that when you take this Enterococcus depleted microbiota and you add back in Enterococcus, you can recover the ability of C. diff to colonize. And so this was starting to get to this hypothesis that having Enterococci enriched in your microbiota might be actually supporting C. diff colonization early and might not just directly be a correlation. Additionally, what we found is that when we added in our lab strain of Enterococcus faecalis, which is strain OG1RF, we found that C. diff produced more toxin. And so this was something that we would repeatedly see, is that the infection looked worse when you exogenously added in this Enterococcus faecalis strain. So this was starting to show us that the Enterococci seem to be supporting C. diff early in colonization. And then when they both get to really high abundances, seem to be increasing C. diff's toxin production and virulence. So potentially having this dual role with both supporting C. diff's fitness as well as potentially increasing its pathogenesis. Additionally, the big question is the Enterococci do really well in the small intestine, but C. diff colonizes the colon. So we wanted to know, do they actually see each other in these circumstances? Is this actually, if you're thinking about, do they interact? And the question is, are they directly impacting each other's behavior? You would hope that they would be localized together. So we did fluorescent in situ hybridization, And indeed, in these infections, we see that the Enterococci, which are the dipococci in the green, and C. diff, which are these purple rods, are completely co-localized, both in terms of the lumen, as well as in these biofilm-like structures up against the epithelium. And so we would find the, C. diff biofilms is a hot area right now, but they've never really been observed in vivo readily, but there's these, ideas that you see these kind of like mats during C. diff infection that presumably would be C. diff biofilm-like structures. And we find that if you image these in our mice, these tend to have biofilm-like structures with Enterococcus and C. diff. And so, stepping back, we wanted to know if we move out of the mouse, can we see if this is a direct relationship? We're seeing enhanced pathogenesis, supportive role for Enterococci. Are they directly interacting with each other? So kind of like thinking backwards, if we step out of the mice, can we see if they're interacting using in vitro systems? And so what we did was we, simply grew C. diff and Enterococcus together in culture. What we found is that when you grow the two together, they don't impact each other's growth rate. If anything, Enterococcus seems to boost how well C. diff is growing in our hands. And so what this was suggesting to us is that they're not competing with each other like a lot of organisms do. And additionally, we found that when they were grown together, Enterococcus was enhancing the amount of toxin production in C. difficile. And so I'm not actually showing those data here, but what I'm showing you is actually the next step, is that what we then did was we grew Enterococcus independent of C. diff, filtered out the cells, and then introduced the cell-free supernatant C. diff. And what we found was actually quite surprising is that still enhanced the amount of toxin that C. difficile was producing. And so when Enterococcus and C. diff are grown together, they seem to work well together, partition resources grow well. And then if you remove C. diff and just introduce the supernatants from Enterococcus to C. diff, it increases this toxin production too. So this is suggesting that enhancement in virulence is likely some kind of soluble factor, some kind of metabolite, something that is signaling to C. difficile to enhance its toxin production. We see this with a myriad of different strains, both our lab strains and clinical strains. We see this with vancomycin-resistant strains of Enterococci, as well as vancomycin-sensitive. And you can see there's faecalis and faecium here as well. And one of the things that's important to note is that faecalis and faecium are actually quite divergent from each other. So, at first we were wondering, is this some kind of faecalis phenomenon where it's like some kind of auxiliary piece of the genome, some kind of, crosstalk between the two that might not be fundamental. But in terms of this phenotype, we find that just about all of the Enterococci perform this action on C. difficile. So it's something maybe core to the Enterococci, maybe metabolism. Additionally, I showed you that we were seeing these biofilm-like structures. So we were interested in biofilm production with these two. So what we did was we again stepped out into the in vitro system and did some biofilm assays with Enterococcus and C. diff. And so we were using some of the classical static biofilms on plastic with the faecalis. And what we found is that when you make a C. diff biofilm, it does make biofilms. And you introduce vancomycin, which is the frontline antibiotic for C. diff, you can kill most of the C. diff. So the C. diff biofilm isn't really providing a robust resistance to vancomycin for C. difficile. However, what we find is that when you introduce Enterococcus faecalis and you make dual species biofilms, that protects C. difficile from vancomycin. And this is specific to Enterococcus because we don't see similar results when we use other biofilm forming organisms like the E. coli. And so what this is suggesting is that, Enterococcus, at least so far, is enhancing C. diff virulence, but it also might be providing a safe haven for C. difficile against the mucosa that might allow it to persist in the face of the frontline antibiotic, vancomycin. What we did in the paper that I'm not showing here is that we went in and we started mutagenizing different aspects of the Enterococcal biofilm formation. What we found is that it seems to be the Enterococcus biofilm that is protecting C. diff because we can reverse this phenotype by getting rid of some of the, or by shifting some of the biofilm structure phenotypes in the Enterococci. So it seems to be the Enterococcus biofilm is encapsulating C. diff and allowing it to persist in Enterococcal biofilm, but we're still working on that pretty substantially. One of the things that's interesting is that we see a lot of C. diff and VRE co-infections, right? Because they seem, Enterococcus does really well in the C. diff infected gut. They have the same risk factors. And one of the areas that we became interested in a collaboration with Derry Van Tyne's lab at Pitt, is looking at horizontal gene transfer between these two organisms. And, we were thinking biofilms, we were thinking, a rich area by which bacteria in the gut might be shuttling around mobile genetic elements. And so we looked at this with Daria's group. And what we found is that if you look at genomes from patients from C. diff and VRE in patients that were co-infected, and you look for evidence of transfer of mobile genetic elements, we saw a significant amount of potential horizontal gene transfer events that are occurring. And so here in each of these, we have blue as the C. diff, and this brownish color is the Enterococci. We find robust transfer of, or predicted transfer of different antibiotic resistance cassettes. So this is TN916. This is actually the transposon we use in a lot of cases to try to move DNA between into challenging strains. So we have a Bacillus subtilis strain that has this transposon. It readily shuttles plasmids over to C. diff. So it's kind of cool to see this actually transferring in nature. And we also saw a myriad of other examples that would happen over and over and over in different patients of evidence of horizontal transfer. But one that was really cool was that we identified this CD0386-like adhesin. And so this is actually a collagen bind or predicted collagen binding protein in C. diff that we were finding in VRE. And so this was really interesting because this was a potential factor that might be impacting how Enterococcus behaves that it might be getting from C. diff. And so we were really interested in potentially, does this impact Enterococcus, kind of the flip side of the coin. And so what we did was we took this this collagen binding protein and we cloned it into our OG1RF strain that does not have that C. diff protein. And what we found is that when you introduce this collagen binding protein from C. diff to Enterococcus, it actually enhances Enterococcus' ability to bind to collagen on plates. And so this was really cool. It's kind of just like an observation. We're seeing potential shuttling of genetic elements between these two organisms. And some of these characteristics that C. diff has, or the protein that C. diff has, can confer a competitive advantage to Enterococcus in terms of binding to collagen, which becomes readily available during C. diff infection. And so just to quickly summarize what I've shown you so far, what we're seeing is that the Enterococci get to high abundances in the gastrointestinal tract. This is leading to an enhancement of C. difficile colonization. So some kind of nutrient gradient here or something like that seems to support C. diff early on. It enhances C. difficile's toxin production, and they make biofilms, which allowed C. diff, we think, to persist in the gastrointestinal tract. But the big question we had was, what is the communication? How are they talking to each other? What is going on here mechanistically? And so we really wanted to kind of know, what is this arrow here that's kind of driving both the supportive aspect of C. diff colonization, but also altering C. diff's pathogenesis. And so the way we went about this was multifaceted. We did some co-culture RNA-seq that I'll show you about, and then we did some metabolomics and imaging mass spectrometry. And so when we put these two organisms together, I mentioned that they don't really seem to impact each other's growth rate that much. We really didn't know what kind of, if we were going to see large changes when we put these two together. But when we grew them in co-culture and then did RNA-seq, maybe not surprisingly, we saw a large shift in the amount in the transcripts in C. difficile. We saw over 300 transcripts that were changed. And if you just look at a heat map, I know that you can't read this, but I've color-coded in red various aspects of C. diff, various genes in C. diff that are associated with amino acid metabolism and dealing with amino acids and metabolizing amino acids. And so what was clearly happening is that when Enterococcus and C. diff were together in culture, C. difficile is increasing the utilization of amino acids. And to further look into this, we collaborated with Jason Pappin's group at the University of Virginia and a really talented postdoc, Matt Junior, who actually did my PhD with in Pat's lab. And we performed genome scale metabolic reconstructions to look at what was happening to C. diff metabolically in co-culture. And what Matt was able to show readily was that when C. difficile is with Enterococcus in co-culture, dramatically changes C. diff's metabolism. C. diff's metabolism is completely reprogrammed. You can see here in this ordination, looking at conserved pathways, we see this large shift in C. diff metabolism. One of the things that was really surprising to us and very surprising to Matt and Jason is that it's predicted that when C. diff is with Enterococcus, it actually enhances its fitness. And so this is a surrogate for doubling time biomass flux. And what we find is that when Enterococcus and C. diff are together, C. difficile seems to be more fit. It grows better in this model. If you look at what factors are largely driving these shifts in metabolism, what you find is that we see large increases in amino acid utilization and amino acid uptake. And so C. diff metabolism is interesting. It has the ability to use, and it primarily uses in the gut, amino acids for energy. So it can ferment amino acids in the gut. And what we found is that a large number of amino acid uptake systems and metabolic pathways were enriched during C. diff infection or in culture when C. diff was with Enterococcus. And so you can see here, one of the highest and most important predictors of co-culture was ornithine flux and then isoleucine flux. And then you can see ornithine can be used by C. difficile to make both alanine and proline, which are the two types of stickling fermentation, and that leads to 5-amino valerate production through the PRD pathway. And then isoleucine can be fermented into methyl butyrate. So that was predicted to be enriched. And so what I'm showing here, you don't have to look at this too much, but C. difficile can make energy through glycolysis or through amino acid metabolism through stickling fermentation. And what the model predicts is that Enterococcus actually drives C. diff to 100% amino acid utilization by providing those amino acids, and glycolysis becomes completely dispensable for C. diff. So C. difficile is relying on likely through cross-feeding or partitioning of nutrients, amino acids when it's with Enterococcus. And so we wanted to see if this was what was happening during infection. And the way we went about that was by looking for these metabolites during infection using imaging spectrometry. And so in collaboration with my good friend and colleague, Boone Prentice at the University of Florida, and his really talented grad student, Jonathan Specker, we did imaging mass spectrometry to look at distribution of a myriad of metabolites during C. diff infection, but specifically the amino acids. And so I think a lot of people might be familiar with imaging mass spectrometry, but MALDI IMS, what we're able to do is take a tissue or a colony, I'll show you both, and mount it on a charged slide, overlay a MALDI matrix, and then you can take a laser and raster the laser, basically pixelating the tissue, which then gets desorbed into a mass spectrometer. You can then get mass charge for each pixel, and you can overlay that back onto a 2D heat map to look at localization of different metabolites and abundance of those metabolites in that 2D space. And so the first thing we noticed when we were doing this was that ornithine, which had been predicted to be one of the most important aspects of interaction in the model, was highly abundant in the lumen during infection. So we almost cannot detect it at all in uninfected mice, but during infection, you see this large increase in ornithine. And what quickly jumped out to us was that we actually saw the exact reciprocal localization with arginine. And so you can see here that ornithine is highly abundant in the lumen, and then arginine is almost completely devoid in the lumen and highly abundant in the tissues. And if you overlay this here, you can see how they perfectly fit within each other. And so here's an H&E stain of the infected tissue. This is the lumen here. This is the damaged epithelium, and then this is edema and underlying lamina propria. So you can see that the ornithine is highly abundant in the lumen. And that was really interesting when we were thinking about enterococcal metabolism, because Enterococcus has the ability to use arginine to make ATP through the arginine deaminase pathway. So it can catabolize arginine into ornithine as a byproduct and eventually into ammonia, carbon dioxide, and ATP. And one of the ways that it brings in arginine is through this antiporter called ARC-D. So this is called the ARC system. And so ARC-D is actually an arginine ornithine antiporter. So it takes in one arginine and releases one ornithine at a 1-to-1 ratio. And so as Enterococcus uses arginine that you can actually see here in just media, it will use up the arginine and then it will pump out ornithine. And so what you can do is you can go in and you can mutagenize this ARC-D antiporter, and you can completely reverse the ability of Enterococci to utilize arginine and export ornithine for anything. And so we wanted to know, this is during these images that I showed you was just during infection in conventional animal, but there's a lot of organisms that have the arginine deaminase pathway. There's a lot of different ways that arginine can be used and ornithine can be used like host. I'm sure a lot of people know about, macrophage polarization and arginine and ornithine. And so we wanted to know if this was driven by Enterococcus. So what we did was we infected germ-free animals with just C. diff. And what we found is that when you just have C. diff, you actually see a high level of arginine in the lumen. And so what we're thinking is that C. diff causes damage to the mucosa and that liberates arginine into the lumen. If you then add in Enterococcus, all of this arginine gets converted to ornithine. And so what we think is that this is relatively specific, especially when you have outgrowth of Enterococci, where the Enterococci are utilizing that free arginine for energy and then converting it to ornithine. And I noted before that C. difficile can utilize ornithine for energy, right? So it can metabolize it into alanine and proline and then ferment it for ATP. And so what we can do is we can quantify this not just by imaging, but by doing traditional metabolomics focused on these amino acids. And you can see this really nicely. That germ-free mice have this basal level of arginine. You add an Enterococcus, it utilizes that arginine and then converts it to ornithine. And then if you knock out the arginine deaminase system, that ARC-D, you completely reverse this. And actually, this was actually really cool to see because it's like a test tube. Like mice are not test tubes at all, but it was actually really cool to be able to see almost the same thing that we were seeing in a test tube, except for in the lumen of mice, in stool of mice. And so the next question we had was, is this the mechanism by which Enterococci might be manipulating C. diff's fitness and virulence, right? Because we know that the ornithine can be used by C. diff. We know that it can be taken up by C. diff. Is that what's going on? And so we went back to our in vitro system and we knocked out the ARC-D gene and we reintroduced the supernatant to see if that's how Enterococcus was talking to C. diff. And what we found is that when you introduce, wild type C. diff to Enterococcus, you see the increase. And then if you knock out the arginine deaminase system, C. diff no longer responds and produces more toxin. So this is what we were expecting. This seems to be the signal that C. difficile is sensing to increase its toxin production. Additionally, if we take germ-free mice and we infect them with wild type OG1 Enterococcus faecalis or the ARC-B mutant, we see a significant decrease in pathology when Enterococcus is unable to produce ornithine through utilizing arginine. So we think that this is what's driving that enhancement of disease severity in animals as well. So the question we had next was, there's two sides to this coin. We have arginine depletion, and then we have the production of ornithine. And so the hypothesis that we had was that ornithine was the driver of pathogenesis in C. diff. It was taking in this ornithine, it was sensing that molecule, right? This is kind of like we were thinking it was direct crosstalk that was driving pathogenesis because you can introduce a supernatants and you see the enhancement. And so that was the hypothesis that I had, that I made Alex pursue for a very long time with no positive data. We actually didn't see that ornithine was impacting C. diff pathogenesis in our condition. We have some collaborators and colleagues that suggest that it might be doing that, but we weren't able to show that here. But what we were able to show is that C. diff seems to be sensing the lack of arginine. And so this had been reported over 25 years ago, that C. diff seems to respond to arginine depletion. And so what we did was we took our, this is again where we introduced Enterococcus to C. diff. If you take this enhancing culture that drives up C. diff toxin and you supplement an arginine, you can reverse that phenotype. So it seems that C. diff is driving up its toxin production by sensing the amount of arginine that's there. Now, one of the things that's interesting, we're still pursuing this about the arginine deaminase pathway in some of these gram-positive pathogens like streptococci and enterococci, is they actually use the ammonia from arginine catabolism to buffer the pH. So this has been shown in some oral streptococci that, as they ferment sugars, because they're lactic acid producing bacteria, they drive down the pH. but they can actually take the ammonia from the arginine deaminase system and buffer that. And so there's actually, I don't know, I'm kind of going off the rails here, but apparently there's in some European countries, in toothpaste, they add in arginine because it's been shown for some of the oral streps that cause caries, that caries are driven by the drop in the pH. So if you add arginine, you can push on the arginine deaminase system and buffer that low pH, right? But anyways, we're very interested in if this is pH driven, but we haven't been able to find evidence that that's the case. So we were normalized the pH and we weren't able to reverse this. So we think that C. diff may be responding to pH as possible. It's been shown before, but we also think that it directly senses the lack of arginine somehow, maybe a nutrient depletion type system, but we don't exactly know the mechanism. We're working on it. But it definitely seems that arginine is the key to the virulence. However, ornithine is not completely ignored by C. diff. As I mentioned, it can take it up. And so what we're looking at here is if you just take colonies and you plot them, you take a C. diff colony and you put it next to an Enterococcus colony without them physically interacting, you can actually see that this is a C. diff colony by itself. There's no ornithine. If you put it in proximity to an Enterococcus colony, C. difficile takes in high levels of ornithine. And if you knock out the ability of Enterococcus to make that ornithine, it completely reverses that phenotype. And what we did was we collaborated with Megan Matthews' lab at Penn in the biochemistry department and made a probe for the proline reductase, which is what ornithine would ultimately be used to make energy. And you can see that when C. diff is with Enterococcus, it dramatically increases the amount of proline reductase activity by taking in that ornithine, breaking it down into proline, and then using that proline for energy. And so we do think that there's a direct role here where C. diff, and I'll show you that one image, then I'll go into the model, but diff is sensing the lack of an organism or the lack of a metabolite as a metabolic cue to increases pathogenesis. And it's also utilizing the other side of that coin, that fermentable amino acid, to then gain a fitness advantage as well. So it's kind of a two-signal system that we think is driving this change in C. diff behavior. And I just wanted to show this. This is an imaging mass spec image of two colonies right next to each other. And so it's C. diff next to Enterococcus here. And I just wanted to show because it shows you how impactful this arginine deaminase system is just on plates. So these are just rich plates. And so this is C. diff on the left and Enterococcus on the right. And so if you look at this is wild type C. diff and wild type Enterococcus, you can see that the Enterococcus is leading to a huge increase in the amount of ornithine that the C. diff colony is seeing, and it's completely starving it of arginine. But if you knock out the arginine deaminase pathway, look how much arginine C. difficile is seeing here. So it's that kind of that cue. It's really, Enterococcus can really remodel that environment, both in the gut as well as just on plates, to completely change what C. difficile is seeing. And so this is kind of like going all the way back to the beginning, thinking about that context dependency. Who else is there can completely change what C. difficile is seeing, and then, of course, remodel the metabolic state it's in. And we know that's so tightly associated with pathogenesis and fitness ultimately. And so what we can do is we can fill in some of these here. So what we think is happening is that when they get to really high abundances, Enterococci utilizes that arginine that C. difficile that is available in the gut. C. difficile is starved of that arginine, is responding to it, but is also using that ornithine to gain a fitness advantage as well. And so finally, what we wanted to be able to do was see if we can reverse this. So what we've been able to show is that if you mess with the Enterococcus metabolism by using genetics, right, you can reverse that phenotype. So we know manipulating the Enterococci can change the outcome of that C. diff pathogenesis. We wanted to know if we could use arginine to do this as well. So if Enterococcus is starving C. diff of arginine, can you use supplemental arginine to kind of overwhelm that Enterococcus arginine deaminase system and alter C. diff, how much arginine C. diff is seeing, and then pathogenesis? And so what we did was we exogenously introduced arginine in the drinking water, and we did not see any difference in the amount of C. diff CFUs or the amount of Enterococcus CFUs in these mice. However, we were able to see that when you added in that exogenous arginine, you saw a decrease in the amount of weight that these mice lost and the disease severity associated with that infection. And importantly, arginine is associated, like I said, with immune function and things like that. We looked at that, we didn't see any differences. What was really cool is that we saw that if you look in the stools of these mice, they had significantly decreased toxin titers. So as you would expect, if C. diff was responding to that lack of arginine by increasing more toxin, what clearly happens is if you overwhelm the system with water administered arginine, you seem to be able to drive down the toxin production in C. diff and ultimately protect these mice from severe infection. So this is kind of the model that we had here, but if you were potentially a reviewer on the paper, you might have asked, what does Enterococcus get out of this, right? And we're very interested in this as well. What we wanted to know is Enterococcus getting anything out of this interaction? So like why are the Enterococci thriving during C. diff infection? And so with the last 2 or 3 slides here, I'm going to show you that, I'm going to remind you that I mentioned that during a germ-free infection without any other organisms, C. diff leads to a large increase in the amount of arginine that's in the lumen. And Enterococcus, we know now, is utilizing that arginine to create ATP, create ornithine. And so you can quantify that here. Germ-free mouse, you add C. diff, you see that increase in arginine, and Enterococcus uses it. So we hypothesized that that might be one of the ways that Enterococcus is getting the arginine through the action of the C. diff toxin, causing disease, damage and disease, potentially inflammation, leads to more arginine. So what we did was we wanted to see if the ARCD mutant had a defect in mice when it competed with commensal Enterococci. So what we did was we took the ARCD mutant and we introduced it to mice that have their own endogenous Enterococci. And then we took the wild type and did the same. And what we find is that early in infection, so day 1 during C. diff infection, you don't see a defect, you don't see a lot of inflammation, and therefore you don't see a defect in ARCD. It can colonize just fine. However, as inflammation increases, and we think the toxin is liberating the arginine, the wild type Enterococcus that we introduced does just fine compared to the endogenous Enterococci, whereas we start to see a defect in the ARCD mutant. So the ARCD isn't doing as well, it's falling out faster. So if Enterococcus doesn't have the ability to use that arginine, it doesn't do as well compared to the endogenous Enterococci that presumably can. And so we think that C. diff is providing Enterococcus with a nutrient that does allow it to thrive, and therefore it's a potentially cooperative interaction here where I drive you to produce more toxin, you remodel the environment with all that damage and disease, I can then use that remodeled environment to thrive. We have also, this is new data here that I threw in as well, that we think that there's other ways that the C. diff toxin is driving a shift in the metabolic environment as well that Enterococcus is leveraging. And so this is heme. And so this is a germ-free animal and a C. diff infected animal. And what you see is that in a germ-free animal, there's not a lot of free heme in the cecum at all. You can't really see it too well. But what you can see here is that when you add C. diff and you cause disease, you see a high level of luminal and epithelial heme release. And so this is something that we actually showed in Eric's lab when I was a postdoc, and we have data from metabolomics in patients as well, that heme is highly associated with C. diff infection. And it's actually independent of just inflammation in general. So patients with IBD have a lot less heme than patients with C. diff. And so C. diff seems to increase the amount of heme that's in the lumen. And one of the reasons why that's really interesting is that Enterococcus has the ability to aerobically respire. It has a heme-dependent cytochrome, but it doesn't make its own heme. So it's really interesting because it has this whole arm of its metabolism, but it needs to acquire heme exogenously. But there's not a lot of free heme in the lumen of the gut. But during C. diff infection, there's a ton. And so what we think is potentially happening is that C. diff causes damage, liberates that heme that can then populate Enterococcus's cytochromes and potentially allow it to grow better. And so to test this preliminarily, Alex has done this. He's done this, I think, 4 times now. But basically what he did was he knocked out the cytochrome in Enterococcus. And what he did was he then introduced these to the mice, just like the arginine deaminase mutant. And what we find is that if Enterococcus does not have the cytochrome, it cannot thrive in the C. diff infected gut as well. And so what we think is happening is that Enterococcus is gaining a competitive advantage in the C. diff gut as well by utilizing all of that damage that C. diff causes, those nutrients that are liberated, to then co-opt and utilize for its own benefits. Okay. And so then we can kind of add this extra arrow that maybe that reviewer wanted, which is, what does Enterococcus get out of it? Well, I think Enterococcus gets a lot out of it because all that damage that's caused during C. diff infection creates this completely different nutrient environment that the Enterococci thrive in. They do really well during inflammation. They do really well when things are perturbed. And so we think that this is kind of a cyclical relationship between the two. And so I'm going to stop there, and I'd be delighted to take any questions. I just want to, of course, shout out our Penn State alumni who drove all of the work, Alex Smith, a lot of former lab members that were intimately involved in a lot of this work. It's a very collaborative effort. We have a large microbiome group, as many of you know, at Penn and CHOP, the Penn CHOP microbiome program. So we do a lot of work with a lot of these incredible members there. I want to just point out Boone and Jonathan, Jason and Matt, and then Daria, who all worked collaboratively on this, and I'd be delighted to take any questions.
Thank you very much, Joe, for that amazing talk. And we're gonna continue with the tradition from last semester. So we ask the first question to be from a trainee, which means a student, a postdoc. So
do those excess arginine that you fed to mice affect the host tissues at all? That could be also affecting the pathogenicity.
Yeah. So can they hear the question remotely, or should
I think people should be able to hear it.
Okay. I'll just try to repeat it as I go too. So is excess arginine that we're introducing in the water impacting the host as well? And so presumably, yes. But what we did was during those infections, we also did flow cytometry to look at neutrophils and macrophage polarization and things that would be classically thought to be associated with arginine and arginase and potentially polarization and potentially impacting the disease. And we didn't see any differences. And although, you'd wonder if it does impact it and how those interactions, and Justin Sonnenberg's group has shown that interactions between ornithine in the diet and the host can really impact C. diff as well. And so it's definitely something that was noted, but what was really interesting, what we were excited to see, was the decrease in the toxin, because that's directly driven by C. diff itself. And so it's like everything, it's probably multifaceted what ultimately leads to those mice doing so much better. But we think that, based on the toxin data, that at least in part due to impacting C. diff. But yeah, it's a really good question.
Thank you.
Thank you.
Any other questions?
So really nice talk. The question I have, it's the biofilm that you were observing in vivo. It really looked well mixed.
Mhm.
It's not that you saw pockets of microcolonies.
Yeah.
So I was wondering whether that observation is reproducible, and then how the way the biofilm develops in vivo, how does that inform that model? Are things coming in and not growing and leaving, or are they actually growing in there, but they're really dependent on one another?
Yeah, so it's a really good question. So the question just quickly is, the structure of the biofilms in vivo looked quite mixed. What's going on in vivo, and are they important early and then transiently what's going on in these biofilms? Working on that quite a bit now. Enterococcal biofilms are interesting, especially in the gut. They've been shown to be small microcolony type foci biofilms, not like a Pseudomonas, big meaty biofilm. And they've been reported to be flat, if you will. And so it's really interesting. We're trying to understand how an Enterococcus biofilm is different from when it's with C. diff, because they clearly are interacting with each other. And if it was flat, how is C. diff getting in there and being protected by that structure? And what about that structure is protective? And those are all things that we're very interested in. And so the in vivo stuff is hard. And it is reproducible. We see these mats in all of the mice up against the epithelium, but it's kind of hard to watch how they grow or things like that. So instead, what we're doing is we're going back to the in vivo model, several different types of models of biofilms, and trying to see if we can note the succession of the biofilm, what's happening long term, and see if we can, and what is the composition of it, and does that change when C. diff is there and when it's not. There's a lot of like kind of other projects in the lab that are associated with, we have a lot of phenotypes that associate with some of the main biofilm drivers in Enterococcus that we have a handle on, but not in the biofilm. And so like, there's a lot of smoke, but we're trying to figure out exactly what's going on. Yeah, so we're not quite there yet, but yeah, thanks.
More questions?
I have a kind of broader one than what you were talking about, but this ability of arginine to, potentially suppress some of the negative outcomes. I was wondering if you've gone back at all to kind of more human data and epidemiological data and actually seeing, do we see less severe C. diff in infections in folks depending on their diets, if we have that information? And like, just how sensitive are we to arginine in terms of infection? Like, is that something we can modulate with diet alone, or would you really need to like bomb it with arginine?
Maybe I should throw this to Jordan, but there's a lot going on with C. diff and diet. There's a lot of very, very interesting work that has come out that's being done that is showing that diet seems to be a really strong modulator of C. diff severity. So that's just one. So I think that diet is one of the low-hanging fruit that we might be able to, if we can understand it, leverage to potentially impact C. diff infection. In terms of arginine and severity, it's a little hard because you're looking for a lack of something, right? And so there's a couple things that we saw in the data sets for this work that was interesting. So one is that ornithine is nicely positively correlated with C. diff, and arginine is correlated with Enterococcus. And we're trying to get at that and seeing if we can use it as a predictor. Going back into larger data sets and seeing if we can see this across a spectrum of age, from children to adults, is things that we're interested in doing. And can the arginine in your diet be predictive of if you were going to get more severe infection, things like that? All things that we have like diet data from this, and we have dietitian people that do stats, and like we're very interested in doing some of that work. It's just harder to directly correlate one metabolite, right? Because people's diets are very complicated, and yeah. But it's like exactly, I think the diet and C. diff stuff is a huge and really exciting area, like, at the forefront of C. diff research.
We have time for one last question.
I was wondering if the heme being released to the Enterococcus and then utilizing it in its cytochromes or whatever, is that potentially also then benefiting C. diff again by like using up bits of oxygen that are around?
Yeah,
or things like that.
That's the hypothesis. It's actually Alex, to his credit, like, in prelims, you do like a project. His second aim was that.
Yeah.
And he's just starting to get to it now, and we're just seeing evidence that it actually does seem to be using the heme. And you would think that like a lot of Andreas Baumer's work has shown this, that the facultative anaerobes utilizing oxygen can make things more like hospitable for the Clostridia that is strict anaerobes. But C. diff is actually quite aerotolerant, surprisingly. And so like, as much as we want that to potentially be that really nice model, I'm not sure it cares that much about oxygen. But what it does care about is heme. And so it has a couple systems that are used to either detoxify or utilize heme, because heme is really toxic to C. diff. And so, and toxic to all organisms. It's one of those like Goldilocks molecules, right? Too much is bad. And so I think that it could be that both the oxygen story, but even if the oxygen story isn't quite as simple as like the Baumler stuff, it could be that just getting rid of that heme is good for C. diff too, because it doesn't want to have to waste energy or get killed by that heme that's there. Because it's kind of cool that C. diff has or drives up a toxic molecule that it then has to deal with itself, otherwise it will kill itself. But that's actually, like, in terms of potentially drugging against C. diff, it's weird that it drives up an increase of heme, but if you can get rid of its detoxification system, maybe it kills itself and you could use it as a drug, which is something that, yeah. Eric's lab has thought about. All right, thank you, everybody.
Thank you, Derek.